normal prostate epithelial cells, as is illustrated here. We don't really know why there is a tumor specificity here, but it may be linked to the expression of proteases by cancer cells versus normal cells. So TRAF6 then mediates the activation of P38 and JNK, as I have told you, and the liberation of the intracellular domain. But there are other signaling pathways which also are linked to invasion: tyrosine kinase SRC and phosphatidylinositol 3 kinase. So we have then elucidated mechanism by which these are activated. And to start with PI3 kinase, this molecule, it's a family of enzymes consisting of regulatory P85 subunit and the catalytic 110 subunit, and they act then on PI45 bisphosphate to make PI345 trisphosphate. And this is a crucial mediator of signaling, and it activates, for instance, the serine threonine kinase ACT, which in turn activates a number of different signaling molecules, leading to a number of effects inside the cell, including cell migration. So how is then PI3 kinase activated by the TGFbeta stimulation? Well, you can see here that the TGFbeta induces ubiquitination of P85, the regular subunit of PI3 kinase. And this is, again, a co-IP experiment in which we have used an antibody against P85 to immunoprecipitate P85, and then an antibody against ubiquitin to blot with afterwards. However, if we knock down TRAF6, there is no ubiquitination of P85, telling us that it is TRAF6 that ubiquitinates P85. So is this of any importance? Well, we localized the ubiquitination sites in P85 to lysine 513 and lysine 519. And if we mutate one or the other, or both, we can see that the TGF-beta induced formation of PIP3, PI3 trisphosphate, which is the product of this enzyme. It's lost if we mutate one, and even worse if we mutate both of these lysine residues. And in parallel, then, activation of ACT, measured here with a phosphospecific antibody against serine 473, and another site that is also important for activation is threonine 308. You see that the TGF-beta induced activation is lost if you mutate both these lysine residues that are ubiquitinated. And is this of any importance for tumors? We looked on material from prostate cancer patients, and we subjected them to staining. As you can see here, more benign prostate cancers versus more malignant one. There is more staining of SMAD2 in the more malignant ones. And there is more staining also for phosphorylated AKT, as you can see comparing these two. Then we looked on whether the activation of AKT was due to TGF-beta induced PI3 kinase activation. And to do that, we used proximity ligation assay, using one antibody against P85 and another against ubiquitin. And as you can see here, small dots that represent that these two antibodies come close to each other are much more prevalent here in the more malignant prostate cancer compared to the more benign ones. So the conclusion is that TGF-beta induced PI3 kinase activity occurs in prostate cancer cells and could contribute to their invasiveness. So this is a summary of what I just said. So after TGF-beta has assembled the heterotetrameric receptor complex, TRAF is involved here, and TRAF then recruits the regulatory P85 subunit and ubiquitinates it on two lysine residues, and then that activates the catalytic P110 subunit, which then phosphorylates and activates AKT, and then causes further signaling inside. So what about SRC? Well, SRC is also an important intracellular mediator, which is activated by tyrosine kinase receptors, like EGF and other tyrosine kinase receptors, and integrins via then FAK. Sarc in turn activates a number of pathways intracellular that leads to proliferation and cytoskeletal reorganization and so forth. And the mechanism then by which TGF-beta activates Sarc was of interest for us. So we could show that TGF-beta do activate Sarc in a kinase-independent manner, shown here using a phosphospecific antibody against Sarc, which monitors activation of Sarc. You see that TGF-beta after 15 and 30 minutes clearly activates Sarc. If you put in an inhibitor for the kinase activity of the receptor, doesn't matter, it still activates. And this is true for one cell, and this is true for another type of cell as well. In this case, we also could show that the TGF-beta stimulation induces a complex between the type 1 receptor and Sarc in this co-immunoprecipitation experiment. If we use an antibody to immunoprecipitate the receptor, V22, you see that in the absence of TGF-beta stimulation, there is a faint band here representing co-immunoprecipitation. But if we stimulate with TGF-beta, this band is more pronounced, telling us that TGF-beta stimulation induces a complex with Sarc. And we could also show that Sarc also binds to the type 2 receptor in this co-immunoprecipitation assay. And here it doesn't matter whether we stimulate with TGF-beta or not. In the absence of stimulation, there is also a co-immunoprecipitation. And in the presence of TGF-beta stimulation, likewise. So this tells us that there is a constitutive complex between the type 2 receptor and Sarc. And we can also show then that the type 2 receptor kinase activity, unlike the type 1 receptor kinase activity, is needed for Sarc activation. Because in this experiment, if we now stimulate cells with TGF-beta, we can see that there is a slight activation of Sarc with this phosphospecific antibody. And if we transfect wild-type type 2 receptor, there is enhanced stimulation of this Sarc molecule. But if we transfect a kinase dead type 2 receptor, we see that there is a loss of phosphorylation of Sarc, telling us that the kinase activity of type 2 receptor is needed. We could then show that the type 2 receptor phosphorylates the type 1 receptor on a specific tyrosine residue, 182. Because if we mutate this particular residue, we see no co-immunoprecipitation we see no phosphorylation of the receptor using a phosphotyrosine antibody of the immunoprecipitated receptor, which is seen for the wild-type receptor as well as for another tyrosine mutation of the receptor. We could also see that after Sarc has been activated, it phosphorylates the receptor itself on several tyrosine residues, as is shown here in an in vitro kinase assay. TGF-beta type 1 receptor phosphorylates Sarc phosphorylates the type 1 receptor. And if you mutate several of the tyrosine residues, it doesn't really much happen unless you phosphorylate 11 of them, then you lose the phosphorylation. Unfortunately, this 11 tyrosine mutant has lost also the kinase activity, so it's not so much useful. But if we mutate instead 8, we can also deprive cells of most of the phosphorylation of tyrosine residues. So we could then show also that the Sarc family members is necessary for TGF-beta induced tyrosine phosphorylation of type 1 receptor, because if we, in this experiment, use now immunoprecipitation of the receptor and the blotting for phosphotyrosine, we see that TGF-beta induces phosphorylation of its own receptor on tyrosine residues, but not only in the presence of Sarc, not in cells that are deprived of Sarc and the cousins of Sarc, yes and Fin. Then there is no phosphorylation of the receptor on tyrosine residues. So what is then the function of Sarc in TGF-beta signaling? Well, in this cartoon, we can see that fibronectin is induced by TGF-beta, which is well known. If you put in the Sarc inhibitor, this is completely lost, as well as it is if we put in a type 1 receptor inhibitor. So this means that Sarc is needed for TGF-beta induction of fibronectin, but not in the induction of another molecule that is also induced by TGF-beta, which is Pi1. We could also show that TGF-beta induces migration. Focus here on the red bars because to make it easier. So TGF-beta closes the wound of breast cancer cells if stimulated for 48 hours. And if one now put in the inhibitor of the TGF-beta type 1 receptor, that it's sort of loss of this effect, and an inhibitor of SRC also gives less TGF-beta induced migration compared to this with these ones. And we can also show that in this mouse embryo fibroblasts that lack the SRC molecule, there is also less TGF-beta induced migration. And if we retransfect SRC into these cells, we can see that we can induce a more efficient migration by TGF-beta. So this is the cartoon which shows us the mechanism whereby TGF-beta activates SRC. So before TGF-beta stimulation, SRC occurs in a complex with the type 2 receptor. SRC consists of the kinase domain as well as an SH2 domain and an SH3 domain. And the SH2 domain is SRC, it binds to a phosphorylated tyrosine in its own tail when SRC is inactive. However, after TGF-beta stimulation, SRC is then, the SH2 domain of SRC is then binding instead to the phosphorylated tyrosine in the type 1 receptor by the type 2 receptor. And this opens up SRC and activates it, and SRC then becomes capable of phosphorylating the type 1 receptor on several tyrosine residues. So this is the model we want to propose. And this is a summary of the pathways that I have described. So amongst the non-SMAD signaling pathways that promotes invasion, TAC1 is involved in activation of P38 and the MAP kinase pathway, as well as PI3 kinase, and in the liberation of the intracellular domain. So this join forces in promoting invasion. SRC, in contrast, is not dependent on TRAF6, but is activated by another mechanism also contributing to invasion, as is ERK, which I haven't talked about. So a few words about epithelial to mesenchymal transition, which is a proteinuria effect induced by TGF-beta. So this shows what it is all about. E-cadherin is a marker for epithelial cells, and upon TGF-beta stimulation, you see that this cuboidal path pattern is lost, and there is less E-cadherin, and what remains is completely different in terms of cell localization. If one stains for actin, you see also the cuboidal pattern, which is changed into more elongated cells looking like fibroblasts. And TGF-beta stimulates this quite dramatically. So EMT is a sort of a natural process which occurs both during the development and in wound healing, as well as in cancer invasion. And it undergoes several stages. So there is a loss of cell contact, and then there is a gain of mesenchymal proteins, and then cells lose grip of each other and becomes more invasive. So in the context of cancer, a few cells in the primary cancer undergo this EMT. These are the red ones here. And then they become more mesenchymal, as is shown here. And then they migrate through the tissue and into blood vessels. And at another site, they extravasate. And again, metastases start to grow again. But then they revert back to more epithelial cells. So this involvement of EMT in metastasis has made us also the question whether inhibition of EMT would be advantageous to prevent metastasis. So we have therefore looked into ways of inhibiting TGF-beta-induced EMT and try to learn a little bit about the mechanisms involved. We know that SMADs are involved as a number of different transcription factors. They are difficult to drug. So what we did was to make an unbiased screen using a chemical library in collaboration with Martino Serial. And this assay was then used in order to try to block EMT induced by TGF-beta. And the assay is displayed here. So this is a staining for E-cadherin, which is epithelial cell stain. And this is in green staining for fibronectin. And after 72 hours of stimulation with TGF-beta, there is very little red stain and very much more green stain. illustrating that cells have undergone EMT. Then, if we now put in the inhibitor of the kinase activity of the receptor, we can block the EMT, so it's reversible. As you can see, the green color disappears and more of the red comes back in the presence of this inhibitor, which inhibits activation of SMADs, but they are also involved in tumor suppressor activity, so it's dangerous to use those SMAD inhibitors in order for cancer treatment. So we then used this assay and we came up with a number of hits, and we could, in collaboration with Andrew Shaw, test 13 of these compounds, and I will just show you data on 2 of them. We call them epithelial plasticity modifier, number 10 and number 13. As you can see here, in this staining for E-cadherin, this cuboidal pattern is completely changed upon TGFbeta stimulation. And then if one put in this EM compound, you see that it's partially reverted, and the other compartment as well. And this is a control using the inhibitor for the kinase activity, which also then partially reverts EMT. So we have some lead substances here which could be potentially useful. However, they need to be developed because the affinity potency of this compound is not good enough. Finally, I'd like to say a few words about EMT, how I can image EMT. So we developed a breast cancer model for EMT imaging based on the fact that we put an RFP, which causes red color, after the E-cadherin gene. And if one do so, the cells become red, as you can see here. If we stimulate the cells by TGF-beta, we induce EMT. The cells lose the red color and become more mesenchymal. And then if we remove TGF-beta, the EMT is reverted to mesenchymal epithelial transition, taking a week or so, and then most of the cells are red. And if we wait another 3 to 5 days, almost all cells are red again, showing us that EMT is promoted by TGF-beta, but reversible. If you remove TGF-beta, it goes back. And this is analysis of certain of the markers. E-cadherin goes down and comes back, and the cadherin of fibronectin of Vimentin goes up and comes down again, as does a number of transcription factors that are shown to be involved in EMT. So I think I'll skip this one. We then tested these cells in sphere formation. So to grow in spheres is an indication of stemless character of cells, and TGF-beta induces EMT, and EMT in turn is related to stemness. So this is illustrated here because if these cells are grown as spheres, TGF-beta induces their proliferation, the size of the spheres. BMP that doesn't do much here, as you can see. These are related compounds to TGF-beta. If one now stains the cells, you see that they lose the red color, which in essence means that they become more mesenchymal, and BMPs have much less effect. If one looks in more detail on the transfer between epithelial cells to mesenchymal cells, the presence of EpCAM is a marker for epithelial cells. And then there is a number of markers for mesenchymal cells, P51, CD51, CD61, and CD106. So by fax analysis, one of our postdocs from Bikohara could show that these cells that are not EpCAM positive, 95%, in the 3D context, without TGF-beta stimulation, most of them did not have any of the additional mesenchymal markers. But after stimulation with TGF-beta, they did so. So a number of them had an additional marker, and several had 2, and a few had even 3, telling us that they have become more mesenchymal. But there is a continuum, and the most sort of invasive cells are actually not those that become completely mesenchymal, but rather those that are somewhere in between. And then we looked at the stemless character of these TGF-beta stimulated cells. And you can see here that if one grows cells in 2 dimensions, in the absence, in the presence of TGF-beta, and then give them additional TGF-beta or not. You see that they are not very red. We can forget about BMP, which doesn't have much effect here. And if one then monitor the stemness properties of these cells, one can see that the control is over here, and this is a dilution of cells, meaning that counting how many, you dilute cells from rather many to almost none, and then you see where you can detect colonies in the various cases. And the steeper the curve, the more stemness character. And as you can see here, the control is over here. If we stimulate with TGF-beta, we come here, and if we then stimulate even more with TGF-beta, both in 2Ds and 3Ds, we come over here, telling us that the cells have really become very stem cell-like and very prone to form metastases. So this is also a finding we made, that TGF-beta stimulates the growth of the mammosphere, as seen here, but surprisingly not invasion, which was a little bit of a surprise to us. So finally, I would like to conclude. TGF-beta antagonists targeting tumor cells, it would be desirable to inhibit TGF-beta's pro-tumorigenic effects, EMT, invasiveness, metastasis, that I have talked about here, while leaving TGF-beta's tumor suppressive effects unperturbed. And this involves growth inhibition and apoptosis. And the approaches one can think of is to inhibit the formation of the intracellular domain of TGF-beta, which I have described. One can also inhibit TGF-beta-induced activation of PI3 kinase, or SRC, or MAP kinases of the P38 junk family, which I have talked about. One can also inhibit AP1 SMAD interaction, which I described. One can also inhibit delta NP63 interaction, which I didn't have time to talk about. Or possibly one can inhibit TGF-beta-induced EMT using the methods that I touched upon briefly. And what will work clinically remains to be sorted out. We are still in the very early phase of these studies. So I'd like to end here by acknowledging the people who have been involved in this work. In my group, these people that are mentioned here, and in particular for what I have told you today, Anders Sundqvist, Ria Vasilaki, Oleg Wojciuk, and Ihor and Maria Jakimovic was involved. And we have collaborated with Marianne Landström and Aris Mustakas, working with us on the non-SMAD signaling and EMT in particular. And we have also collaborations with Kohei Miyazono in Japan, Peter Tendaike in Holland, and Mitsuyasu Kato in Japan as well. And those marked in green have left the laboratory and work elsewhere for the moment. Thank you very much, and I'd be happy to answer questions.
Thank you for that very interesting talk, Dr. Heldin. So the question I have is this EMT transition that you spoke of, which can be triggered by just TGF-beta cells. Is that the only pathway for EMT transition? In other words, if you inhibit the TGF signaling, is there an alternative rescue pathway by which this transition can occur? After all, the cancer cells do compete for growth with the normal cells.
This is a very good question, and the answer is that there are also other signaling molecules that promote EMT, and in particular, growth factors that can activate RAS, the oncogene RAS. For instance, FGF promotes EMT as well. And in the context of RAS activation, which is very common in tumors, the TGF-beta effect is much stronger. There is a sort of collaboration between RAS signaling and TGF-beta signaling, which is particularly efficient in inducing EMT.
So, sorry to follow up. If you inhibit the TGF signaling, does the RAF signaling then get more activated? And also, what are the other factors which would enhance the kinetics of TGF signaling? I mean, is it related to the other physiological factors.
So enhanced beta signaling?
Yeah.
Yeah, that is brave, I would say. The TGFbeta has so many unwanted effects, so I think that would be dangerous.
No, I'm saying what are the factors which enhance the transition? You showed different slopes of transitions, right? So
that's an important question, of course. TGFbeta is overexpressed in cancers, and the question is, what is the mechanism behind the overexpression of TGFbeta in cancers? That is not so well studied, and it's not known exactly what is the mechanism that induces TGFbeta. But it is seen that several of the other cytokines and growth factors can do that. So it's possible that in the milieu in the solid tumor, where a lot of chemokines and cytokines and growth factors are produced by cancer-associated fibroblasts, immune cells, and cancer cells themselves, that can contribute to the increased amount of TGF-beta seen. And the TGF-beta in tumors can be made both by the tumor cells and by the stromal cells.
Thank you. Thank you, Professor Kaul. This is Karthik here. Hi. Here. So, TGF, does it have a concentration-dependent effect, like the TGF-beta? The cytokines usually have a concentration-dependent effect.
Can you speak louder, please?
Cytokines usually have a concentration-dependent effect, right? So the TGF-beta, does it have the signaling pathway that TGF-beta chooses? Is it concentration-dependent?
Sorry, I cannot hear the question.
So the TGF-beta, the cytokines are usually concentration-dependent, right? So the pathway that TGF-beta chooses, is it concentration-dependent?
If you want me to rephrase it, what he wants to know is whether TGF-beta activity or is concentration-dependent. Certain concentration it chooses certain downstream effect, certain concentration it chooses different downstream effect.
TGF-beta is a very potent molecule. It acts at sub-nanogram per ml concentrations and gives quite a robust response for epithelial cells. There is a need for a little bit higher concentrations to act on fibroblasts, where the different concentrations, in addition, induces different signals. It's less well studied. And I don't think there is any solid evidence that that can happen, whereas there is a lot of speculations.
Thanks. So follow-up question is that, so in a fibrosis environment, we have a higher TGF-beta microenvironment compared to a normal state. So how does the signaling differs on both the sides?
Yeah. So in a fibrotic environment compared to normal environment, we'll have higher TGF-beta. How does that affect the signaling? Yeah,
Yes.
Is that correct?
Yes.
It is true that TGF-beta induces fibrosis very efficiently because of its induction of extracellular matrix molecules. It is also true that in this sort of extracellular matrix, the stiffer it is, the more collagen bundles it has, the more TGF-beta will be activated, because it is activated in an integrin-dependent manner, and integrin then sort of acts through this collagen pathway, collagen fibrils. And also stress that TGF-beta, as I told you, is made as a latent inactive precursor, and it needs to be activated before there is any effect on cells. And there is quite a lot of TGF-beta laying around in our tissues that are inactive. So if one wants TGF-beta activity, the fastest way is to activate what is already there out in the tissues.
Yes. Thank you.
Thank you, Professor. That was a great overview of TGF-beta activity. So I have two questions, if the time permits. First one is, your initial, when you expose TGF-beta for short time, longer time, you had a different signaling pathway altogether when you expose longer, isn't it? So that is I was kind of intrigued by that, because you know how TGF-beta can be pro-death in the beginning and then more pro-tumorigenic later. So does that depend on kind of constant exposure to TGF-beta, or can you have a cellular system where it didn't see TGF-beta at all, but then at the later stage when you expose to TGF-beta, it is still being pro-tumorigenic? Or does it need TGF-beta to be present from the beginning?
Well, I think the answer is that initially TGF-beta induces a number of genes, including several transcription factors, which in turn activate other genes. And that gives rise to the very different pattern of signaling pathways that are activated at the later stage. But in a tissue, we have to think of this as a continuous process where TGF-beta is around all the time. And that would then sort of probably lead to that we will get this effect that we see after prolonged activation in vitro. And that will also be what is mediating the effects of TGF-beta, tumor suppressor or tumor promoting.
Yeah. So with your work on SRC and other PI3 kinase, where you show that TGF-beta, for example, SRC is required for TGF-beta activation. So can we use SRC as a prognostic marker, for example, to treat these patients with the TGF-beta inhibitor?
That is certainly a possibility. I should say that SRC, as you all know, is the first discovered oncogenes and received a Nobel Prize in 1989, going to Varmus and Bishop. And SRC is certainly involved in many tumorigenic contexts and activated not only by TGF-beta, but by tyrosine kinase receptors as well. And it could certainly be a marker if you found a good reagent to monitor phosphorylation of SRC specifically, which is theoretically possible with the phosphor-specific antibodies available, but in practice it might be challenging.
Thank you, Carl, for highlighting the latest developments on TGF-beta signaling. In your slides, I saw that PI3 kinase activation, you are calling it as non-SMAD, right? But in prostate cancers, I see phospho-SMAD2. How to understand that it is non-SMAD?
Well, TGF-beta induces both SMAD signaling
Correct.
and non-SMAD signaling. So if we want to monitor TGF-beta signaling, we can monitor both. And that will give us an indication whether TGF-beta is active or not, as was the case in the prostate cancer cells. So in the more advanced cancers, yes, there is more SMAD activation than in the more benign ones.
So if we prevent that SMAD phosphorylation, do you still see PI3 kinase activation?
Yes, yeah, we would, because if we inhibit the kinase activity of the type 1 receptor, we will completely lose SMAD phosphorylation, SMAD activation, but we will still retain PI3 kinase activation.
Okay. That, only then we may be, we can ascertain that it is non-SMAD pathway, right?
Yes, but I think your point is well taken, because it's often so, which I gave an example of, that SMADs collaborate with non-SMAD signaling. So it's not so separated, it's sort of crosstalk.
Hi, Carl. Thank you for the fascinating talk. My question was, when the TGFbeta is being secreted, can you give some perspective on how it auto-stimulates the cell towards a proliferative program? And if there is some kind of positive feedback that keeps going on, and is there a way to break that positive feedback loop?
So can you repeat the first part of the question?
When TGF-beta is being secreted by the cells,
TGF-beta is secreted?
right?
Yeah.
Can it auto-stimulate the cells
Yes.
towards a positive feedback loop?
Yeah.
And is there a strategy to break that loop?
No, this is quite possible. The autocrine stimulation has been described both for TGF-beta and for other growth factors, PGGF, that we have worked on ourselves. So that's certainly a possibility. In the case of PGGF, there is a tumor type called dermatofibrosarcoma protuberans, in which the PGGF chain gene has been hooked up to, fused with the collagen gene, and these cells make enormous amount of this fusion protein, and then the growth factor part is cleaved off and acts in a truly autocrine fashion, driving tumorigenesis of this particular tumor. And the similar situation do prevail for TGF-beta. Autocrine stimulation certainly occurs.
One more question. When you are applying all these EMT antagonists, you are creating cells with different kinds of hybrid states, which are neither fully epithelial nor fully mesenchymal. And as you said, some of them have higher invasive potential than a fully mesenchymal state cells. So in terms of clinical therapy, when we are applying all these antagonists, are you not increasing the intratumoral heterogeneity of your population? And
Yes.
What is your idea?
No, you're right. When we started this project, EMT was sort of thought of making epithelial cells more mesenchymal, and the more mesenchymal ones would be more invasive. So blocking that transition could potentially prevent metastasis. Now we have learned that it is more complicated, because it's not the fully mesenchymal cells that are the most invasive, it's those that are in between, partial EMT. And then, of course, it becomes more tricky to sort of find an appropriate way of using TGF-beta inhibitors in order to prevent EMT and also prevent metastasis through this effect. But should also be aware that the growth of metastasis at the distant site is dependent on MET, making the mesenchymal cell back to becoming epithelial. So that context, TGF-beta inhibitor could also be not so good. So quite frankly, I think it will be difficult to use TGF-beta inhibitors to inhibit EMT. Probably inhibition of the other protein pathways or local inhibition to prevent the effect on the immune system or on the cancer-associated fibroblasts or blood vessels would be more efficient.
So I had a question. So my question was again a bit more on the inhibition of receptor 1 induced EMT. So I wanted to understand a bit more on how localized inhibitor therapy is, and if receptor 1 inhibition is more of a therapeutic treatment or a preventive measure that is taken against tumors.
I think it would be very dangerous to use these inhibitors for prevention, because that could promote formation of tumors. Particularly colorectal cancer seems to be very much, in that context, TGF-beta has a very important suppressing effect. And if you then mutate molecules along the TGF-beta signaling pathway, you actually increase the risk of getting colorectal cancer. I think that would be very dangerous. The problem is that TGF-beta has this dual effect. First, it suppresses in the early stages, and then it promotes in the late stages. And if we are going to use inhibitors for treatment of patients, we really need to be very cautious not to remove also the suppressor effect.
A follow-up to that, so how localized exactly is the inhibitor therapy? Because if it was not very localized to tumor sites, it would inhibit EMT across the body as well, which has plenty of negative effects as well, right? So how localized is the current therapy?
Right, EMT is important in wound healing, so that would be a potential side effect, that you would perturb wound healing. Whether that would sort of be a problem, I'm not so sure, because there is so many other molecules involved in wound healing, so maybe it would not be so disastrous. But I think there are other problems with inhibiting EMT, as I just said.
Sir, excuse me, sir. Sir, thanks for the interesting presentation, sir. This question might be out of curiosity. That serine threonine kinase you mentioned about, no, sir, in the beginning of the lecture, and tyrosine kinase, serine threonine kinase, is that what's the specificity of choosing a single amino acids like lysine mutation and tyrosine phosphorylation? Would it lead to like side effects, sir, like adverse side effects?
Can you say that again?
Sir, what is the significance of choosing a single mutation, sir, like single amino acid mutation? In one of your slides, I have noticed like lysine have been mutated to arginine, I guess.
Well, the type 2 receptor of TGF-beta is a serine threonine kinase receptor. It phosphorylates the type 1 receptor on serine threonine residues. But what we found is that in addition, it can phosphorylate a tyrosine residue, a specific one, 182, and that leads to activation of SRC. Okay. But I'm not sure that I answered your question.
So my question is, sir, what kind of amino acids we should target while making it inhibit certain certain receptors?
Okay. You mean which of the tyrosine residues that are phosphorylated by SRC that we should inhibit?
Yes, sir.
This is an excellent question, and this is exactly what we are now working on, but we don't have any data yet.
Okay, so I'll follow up your research paper, sir. Thank you.
Thank you.
Right. Thank you, sir. Thank you, Professor Heldin, for that exciting and insightful talk. It is amazing and daunting at the same time to hear the nuanced approach necessary to develop therapies in cancer. Yeah. Once again, thank you, sir. I'm sure this. Yeah, please. You want? I'm sure this session will leave a lasting impact on all of us. Now I'd like to invite Mr. Vinod from the Global Engagement Office to deliver the vote of thanks.
Thank you very much, Professor Santosh. It gives me great pleasure to offer my vote of thanks on this special occasion. Thank you, Professor Carl-Henrik Heldin, for the insights on cancer and the activity of TGF-beta. It was really fascinating. And thank you for accepting our invitation to visit us at IIT Madras and Chennai. We have been humbled by your kindness and professionalism throughout our interactions. We are truly grateful to have you here today. We must also thank the IIT Madras leadership, with special thanks to Professor Kamakoti, Director IIT Madras, for conceptualizing and fully supporting the lecture series. Professor Raghunathan Rekaswamy, Dean, Office of Global Engagement, has been our guiding light. Our heartfelt gratitude goes to Professor Mahesh Panchadmula, Dean, Alumni and Corporate Relations, for graciously accepting our invitation and delivering the opening remarks. We sincerely hope that the interaction of biotech department faculty with Professor Heldin have sown two seeds of curiosity and inquiry. We appreciate our faculty members' help and cooperation throughout this process. I also express my thanks to the staff of Global Engagement and Alumni and Corporate Relations for the support given in organizing this historical event. My sincere thanks to Professor Barun Sarkar from Mathematics for kind enough to spare this hall when we approached him. Finally, I would like to thank the audience for their insightful questions, and we would like to thank Professor Heldin again for his patience in answering all our questions. With this, we conclude today's session, and we invite you all to the Professor Heldin's talk tomorrow morning at 10:30 a.m. at IITM Research Park. Thank you once again, and have a good day. Thank you.